Okay. So, hi everyone, I'm Flavia and I am a graduate student here. I'm working with Dr. Vincenza Colonna, she's sitting there. And today I want to explain to you what I have found in rats using pangenome graphs and genome-phenome analysis. So I think the strength of this study is the use of the pangenome and the use of HX-based rats. For this thing, I want to explain to you a little about the pangenome, but I am not going inside because it's too much for you at this hour. So the first question is that what is a pangenome? So a pangenome is a collection of genomic sequences. As you can see here in this figure, both of these are an example of a pangenome. The first one you can see these are unaligned sequences. Instead, in the second one, you can see there is an adding, so the sequences are aligned. Both of these are an example of a pangenome. It means that the pangenome is not tied to graphs, for example. So the other question is that what are the components of a pangenome variation graph? To build the pangenome that I use in this analysis, I used this tool that we developed, PGGB, Pangenome Graph Builder. This tool gave us this type of pangenome variation graph. And the first component that is really interesting are nodes because that represented the sequences in the graph. Then we have links that are represented here as arrows and connected all the nodes. But in that case, the important thing here are paths because the paths here explain it to us how the sequences goes through the graph. And here you can see two different genomes. As you can see, the node 1 and node 4 are the same for both genomes. What changes here, the node 2 and node 3? This is a representation of single nucleotide polymorphism, and this is the way in which we represent the variation in a graph as a bubble, as you can see then in the other slides. So obviously the question here is that why do we need a pangenome? So we need a pangenome here because with genomic standard approach, the idea is that all sequences are aligned against the reference genome. And also when we have a new sequence, also this sequence is aligned against the reference genome too. So in this way, we are able to see only the difference that there are compared to a reference genome. Instead, with the pangenomic approach, all sequences are aligned against each other. And also, obviously, when we had a new sequence, also this sequence is aligned, as you can see here, to the others. And this way, we are able to capture all possible variations and in particular structural variation. So what we have done so far, this is the you can see here the complete sequence of a human genome. So thanks to this scientists, we have the first telomere to telomere genome assembly. And we used in this work our tool PGGB to build a pangenome using the 47-year T2T assemblies. And we are able to fix a lot of complex regions that we never seen before. So we want to do the same with the model organisms, and we started to do that with the mouse. So we are doing a T2T of one parental, DBA2J. But for now, I want to explain to you what I have done using HXB rats. So as you know better than me, this is the family that I used. All starts when they done a breeding between ASHR and BNLX. The idea here is that to have a recombinant inbreed rats family. The powerful of this family is that we can do genome-phenome analysis, and we have already in GeneNetwork a lot of AQTL already mapped. And we have this data obtained by 10x linked reads technology. So before starting pangenome, and the guys before me explained to you what is meaning. So the idea here is that we have the presence of Busco genes. Here I use the mini complexum. I don't know if it is the right pronunciation. And here you can see stayed on Y axis all the samples that I used. So the idea is that you can see that the majority of assemblies have the complete and a single copy of Busco genes. and this is important for this sample in particular that I use to validate my results that I will show you. So what I have done, this is a pangenome of chromosome 12. I explained to you only this one, not for all chromosomes. And the idea here, you can see that each line corresponds to a different sample. Here you can see the reference genome. And the vertical lines correspond to variation in this graph. Not see the color here because these are only randomly. But the thing that I want to show to you here, it's the difficult to build also centromeric region, because you can see here that I had a great problem to add, to rebuild the centromeric regions. This happens because the sequence that I used, the contigs are too short to map very well in that region and rebuild the centromeric region. So this is the total length of the pangenome, and this is instead the length of chromosome 12. So as you can see, the length is bigger because, as I told you, I am able to capture all the diversity in that chromosome. So I started to study a little microvariants, and what I found is that you can see here the count of variations for genomic standard approach and pangenomic approach. And you see here simple variations. For simple variation, I mean SNPs, indels. Instead, for complex variation, I mean combination of variants, as you can see here. And so the pangenome is able to see complex microvariants that are seen a little in the genomic approach. And what I have done is annotation of these variants. You can see here the annotation of these variants by impact. You can see here high, low, moderate modifiers. So the first thing is that the majority, as we expected, have modifier impact, but the message here is that we have annotation also for complex microvariants, so it means that maybe these are real. So the other thing that is really useful to do is that to check if these variants called by the pangenomic approach are real or not, is try to compare these variants against the genomic standard approach, because this is my true set. And for this thing here, you can see this comparison. This is the precision and the sensitivity for all chromosomes. And you can see here the precision, for example, for all variants, indels, and SNPs. The colors meaning the easy region of the genome and the hard region. In the easy region, I try to remove the complex repeats, complex rearrangement that we know by now. And for our region, instead, I considered the whole genome. And as you can see, for SNPs, in particular for the easy region, the precision is really high, is near to 100%. In the hard region is near to 90%. Obviously, you see that this decrease for indels, but this is still a problem also to study indels in the genomic standard approach. Obviously, my idea is to try to identify new variants called only by the pangenomic approach. As you can see here, the majority of the variants present in the pangenome are present also in the genomic approach. Is that what I expected? But I want to focalize on that type of variation. And before do the validation of that variants, I try to see if I see any abnormal trends when I considered, for example, the variants that are called by only genomic approach and only pangenomic approach. Here you can see the allele frequency spectrum. This is a count of allele frequencies. And you can see that for both of these methods, the trend is the same in the easy and in the hard region. And also the length of the index is pretty close the same. So it means that I didn't see things that maybe can corrupt the variant caller steps. So for this thing, I focalized on only variants called by the pangenome. These are the type of variants that we have here. And I focalized my attention only on SNPs. These are the number of SNP discovered. So what I have done to see if these variants are real or not, I try to map the raw reads of SHR samples against the pangenome to see what I found. This is the number of the variants that are real. And then I chosen randomly 25 variants that are near or inside the repetitive regions. And I validated the 12 variants that are or inside or near the repetitive regions with the Sanger sequencing. And these are the variants that are present in that genes. This is the impact of the genes that are modifier, and only one is low, and this is present in the P arm, so this difficult region of the genome. So we have these variants. What's next? I try to do the GWAS. The difference between GWAS and FWAS is that in the GWAS, I try to see the association between one phenotype and many genetic variants. Instead, with the FWAS, I try to see if there is an association between one genotype and many phenotypes. For this thing, I used the variants that I validated and the phenosome that I downloaded by gene network. So in the end, I find that one novel SNP is associated with two different traits. This is the phenotypes and this is the LRS. Here, I focalize my attention on these two different peaks. The first one is this is an association with the chromogranin expression in hippocampus. This is a protein that is expressed by endocrine cells, and this is used also as markers in many tumors. But the one that significantly and I like is this one, because this SNP is associated with this insulin glucose ratio. And I see in another study that they found a near association at this position, so pretty close to us with the insulin resistance. This is what happens with another validated SNP. As you can see, not all is good because, for example, in that case, you can see that the p-value is not significant with these SNPs and insulin concentration, but I found this association with our marker and blood glucose. And I see that they done also in another study. They try to do to quantify the faster glucose, and they identify a locus pretty close to our locus that is involved in diabetes using a rat model. So in the end, I want to explain to you also a little what I've done about the structural variants. For structural variants, I mean variants that have length measure of a 50 base pair. And we try to do this analysis using 4 different methods. The first 3 are assembly based, and the last one is PGGB, our tool. So the majority are called by all the methods. But to do a filter, I try to see the variants that are seen by at least 2 tools in SHR sample. Then I try to see only the variants that have high impact, that have these sites. And then I validated with the nanopore sequencing 6 variants. Of these 6 variants, 4 are deletion and 2 are insertion that have high impact. For this session, I want to highlight all these one. I know that maybe you are interested in the others, because in the others I see only a difference between male, female, and so on. But here I want to show you this because I see a strange thing. So this structural variation, that is a deletion, is present in all HXBs, but is not present in the reference genome. And for me, this is really strange because the majority, there are some strains that are pretty close to the reference genome. So the idea is that what happened here, maybe there is an error in the reference genome that we don't know. And this is an example. This is the structural variation that I extracted from the graph genome, pangenome. And this is the location of this deletion that is present in this gene that is associated with linked with Parkinson's disease, Alzheimer's disease, so many neurological disorders. So in the end, I want to thank all of my group and thank you for your attention. So if you have any questions, I am here.